Hello, class. I'm your biology teacher, and today we are going to explore enzymes, the molecular machines that drive life itself. Think of enzymes as expert workers in a factory, each specialized in speeding up specific tasks. But how do they work? Why are they so effective? And what's the secret behind their structure? Let's dive in. First of all, we will discuss what are enzymes. Enzymes are actually proteins that act as biological catalysts. They speed up chemical reactions without being used up. Imagine trying to assemble a piece of furniture without a screwdriver. The screwdriver makes the task faster and easier, but it does not get used up in the process. Same goes for enzyme. Enzymes are incredibly specific. Just as a key fits only one lock, each enzyme targets only a specific substrate. Their specificity is vital for life processes, from digestion to DNA replication. Our next topic is structural component of enzymes. Now, let's talk about the structure of enzyme. Enzymes are complex molecules that do not work alone. They often need helpers to do their job effectively. There are several important components that make enzymes so efficient. For example, apoenzyme, holoenzyme, cofactors, coenzymes, and prosthetic groups. First of all, we will discuss apoenzyme and holoenzyme. Apoenzymes, this is the protein part of the enzyme, which by itself is inactive. Think of it as an engine of car without the other parts. It's not going anywhere. Holoenzyme, when the apoenzyme combines with its helper molecule, it forms a functional enzyme called a holoenzyme. This is the active form of enzyme which is ready to work. Okay, so we will say holoenzyme is the complete enzyme made up of both protein part and non-protein part. For example, the apoenzyme is like a screwdriver with no handle, while the holoenzyme is fully functional screwdriver ready to turn screws. Next, cofactors. Many enzymes need a non-protein molecule to help them perform their job. These are called cofactors. So basically, cofactor is a non-protein molecule of an enzyme. It can be metal ions like zinc, iron, magnesium. For example, hexokinase, an enzyme involved in sugar metabolism, need magnesium ions. Magnesium ions are here cofactor, which are helping hexoenzymes in sugar metabolism. Coenzymes. What are coenzymes? Now, sometimes the enzyme needs a larger organic molecule to help out. These are called coenzymes. Typically, coenzymes are vitamins or their derivatives, like coenzyme A, NAD, which helps in cellular respiration. Moving forward, we have prosthetic groups. Some enzymes have an even tighter bond, or you can say covalent bond with their cofactors or coenzymes. These are called prosthetic groups. Unlike coenzymes that leave the enzyme, prosthetic groups are permanently attached to enzyme. Think of them as the glue that holds everything together. Try to understand this with an example. For example, heme in a hemoglobin molecule is a prosthetic group. It binds to the enzyme so hemoglobin can carry oxygen in the blood. Next is activator molecules. In addition to cofactors and coenzymes, enzymes can also be regulated by activator molecules. These small molecules enhance enzyme activity, like adding fuel to fire. Activator can change the enzyme's shape, making it easier to bind the substrate or increasing its overall efficacy. For example, calcium ions can act as activator. Moving forward, we have two models which explain enzyme activity. These are lock and key model and the induce fit model. What is the lock and key model? This model suggests that enzyme is a rigid structure and only able to bond with substrate that fits its active site exactly. This suggests that the enzyme's active site has a shape that perfectly matches the substrate, like a lock fitting a key. And the second model is the induced fit model. This model says that enzyme's structure is a bit flexible. When the substrate binds, the enzyme's active site changes shape slightly to better fit the substrate. You can think of it as an elastic band stretching to hold a ball more tightly. Let me give you some real world examples. For example, amylase in your saliva breaks down starch, needs chloride ions as cofactor. Similarly, DNA polymerase, which is involved in DNA replication, it needs magnesium ion as cofactor. Moving forward, we have lactase, which helps break down lactose in dairy products. We can say enzymes are finely tuned machines that need the right tools, cofactors, coenzymes, prosthetic groups to do their job efficiently. So, enzymes are far more simple. They are dynamic, multi-component molecules that needs their cofactor, coenzymes, and sometimes even activators to function. They are truly the unsung heroes of biology, enabling reaction to occur quickly and efficiently. From digestion to DNA replication, enzymes are driving force of life. Remember, an enzyme is like a factory worker who needs all the right tools to get the job done. Whether it's an apoenzyme or holoenzyme, a cofactor or coenzyme, everything works together to make the life possible. Without enzyme, there would be no life as we know it. So let's appreciate these machines, molecular machines, and all the cool components that help them to do their job. You can ask any question to me. I will read all the comments and will answer you individually. So, in the next class, we will discuss different characteristics of enzymes and the mechanism of enzyme action. Until then, goodbye, class.